All right, welcome everybody to the BioC 2021 Lightning Talks. I'm sure you've heard this a million times before, but if you have questions for the speakers, please put them in the Q&A tab. Because we have multiple speakers, if you can write the speaker's name in your question, that would be great too. We may do some questions in between talks, but most of the questions will be done at the end. You can also use the chat, and if you'd actually like to ask your question on stage, you can raise your hand and we'll bring you on. So at this point, I'm going to hand over the session to Martin Morgan, who will be chairing this session.
Thanks, Erica. It's great to be here and to be able to moderate these lightning talks. They're my favorite part of the conference because just about anything can happen. And, you know, I personally am extremely disorganized, and Erica's extreme organization only partly compensates for that. So I'm really looking forward to everybody's presentation and participation, and the Q&A place is the place to go to ask questions. We've got 6 speakers. They've got 4 minutes each. They're in charge of their own, getting their own slides onto the screen, so that should be fun. And our first speaker, I'm just going to start my phone here. I'll forget to start my phone several times, so some people will be lucky. But Angie Zhu is going to talk about R-empowered large-scale genomic research and novel drug development. So I'll turn it over to you.
Thanks, Martin. Let me share my screen. Okay, I hope everyone can see my screen okay. Okay, that's great. I'm Angie. I'm a computational biologist with 23andMe. Today, I'll briefly introduce how we use R to empower large-scale genomics research and novel drug development in 23andMe Therapeutics. Also, most importantly, we are hiring for the 23andMe Therapeutics computational biology team. So if you're interested, please feel free to reach out. Here are my contact information. 23andMe is a genetic-based biotech company. Our mission is to help people access, understand, and benefit from the human genome. There's 3 parts of the business. One is the consumer, where we have the customer genotyped, and we have a large database of these customers that are recontactable from our daily survey. Really, customer is the core of the business at 23andMe. And then among this more than 11 million of customers, more than 80% of them opt in for research. So that is the second part of the business, where we use this phenotypic plus genotypic data to do basically large-scale GWAS studies. And then with that, we actually can benefit from the data from our customer and pay the customer back by looking into drug discovery and also provide novel consumer products that comes from this research results, and then we pay them back to the customer. So with the thought of benefit our customer from this large genetic data set, in April of 2015, 23andMe Therapeutic Initiatives was founded with a focus on discovering novel treatments for patients with unmet medical needs. So far, we have more than 100 employees in the South San Francisco site. We are fully functioned for drug development and also vet lab experiments and stuff. I want to highlight that one of the founders of 23andMe Therapeutic is Robert Gentleman, who is the co-creator of R, also founding head of Bioconductor, and also former head of Genentech.com Bio. With that, I would like to say that really computational biology team at 23andMe Therapeutics is the leading power of drug discovery. We are redefining healthcare because we have this large scale of data set. This is really the world's largest recontactable database with the phenotypic and genotypic data. And so far, we have about more than 40 programs that are under development in therapeutics. And most importantly, we largely shortened the time that needed for drug discovery. For example, our CD96 program that is collaborated with GSK, we only use about 4 years to find this target, comparing to around 7 years, which is the normal time that used in the industry to find the drug target. So there, on top of the genotypic data, genetic data, from there to find the best drug targets, actually there are a lot of other informations that are needed to add on top of the genetic data that we have. One of them is the Rna-Seq data. So here I'm showing a pipeline that is implemented in the 23andMe therapeutics to use Rna-Seq data to find informative signals. Okay, thanks. Okay, I'm out of time. So yeah, sorry. Yeah, okay. So basically, all the green blocks are the R packages we develop internally, and yeah, sorry, I'm out of time, but that's about it. Thank you.
Excellent. Thanks very much. That's what I like about these lightning talks. Really put the pressure on. Next speaker, getting himself set up, I hope. Jian Hu will talk about updates on the RCWL project. Over to you.
Okay. Yeah. Hope you guys can see my screen, right? Okay.
Yes.
I can. Great. Hi, this is Qiang from Roosevelt Park Comprehensive Cancer Center. We have a bioinformatics core team working on all kinds of like bioinformatics tools. So today I'm speaking about the Icw project, some update. First, what is Icw? This is our interface to the common workflow language. We have two core packages for this project. One is Icw, another one is Icw pipelines. This one is a collection of all kinds of like a portable, modularized, and customized for tool and pipelines. Now it's almost 200 tools, all writing as Icw. And this tool is kind of like the most, some of the major features like work in different platform in high performance computing system in the cloud. Like we daily use this daily for our data analysis, and also this is seamlessly integrate with the R bioconductor packages. So like every function or tools for bioconductor can be easily to be converted to a CW tool, and also any CW object can be built to a Cni automatically. So for user only can, for user, you just like update your package and search what you are trying to run and load the package to our environment, assign value to the object, then just run it in different platform, and that's it. For developer, you can use the Icw package to build a new tools to build a workflow. So we have a home website for this package, icw.org. So we have some document and some tutorial for this package. And this year, we also published a paper in bioinformatics for this package. So here, actually, this is some update for this project. We have this is a GitHub repo for this project. We are moving the CW version from 0.1 to 1.2, and we are going to add more features, and of course more tools will be added to this package and more documents. And for now, we have a lot of tools and pipeline integrated to this package, and we have a recipe to store all this source code for all the tools, and you can check how this tool been writing, and it's stored in Icw and CW format. So basically, you can use Cdw format directly. Also, we have another package. It's called a Cdc Client. This one is a client for the Cdc. Cdc is kind of like Untitled. It's just powered by Cdw, by the cancer genome cloud. It's powered by 7Bridge actually. So this is one compatible to Cdw. So we have some major functions developed like authorization and file management, and most importantly, we can add any Cdw object to this cloud. So basically, for the Cdc cloud, we can run any tools you want. Just use this function to upload the tools, the pipelines to the cloud, and build a new task and run the task. That's it. It's like a sublet job to a high-performance computing system. So it's very, very simple. We have a big project running in Cdc now. So basically, this tool is in active development. So we also had two workshop last year. Okay, that's the update. Thank you. Welcome to submit any kind of comment or suggestion. Thank you.
Thanks, that's great. And our CWL is a great project. Our next speaker is Eric Davis, generating covariate matched ranges with null ranges and visualizing them with Bento box. Sounds like a good dinner time talk. Perfect. I think we've got your screen, but we don't have any sound. Still screen and no sound.
Hello.
There you go.
There you go.
Your sound
Okay.
anyway.
Sorry, I haven't had a chance to test this out yet. Let's see. Sharing my screen. Cool. Okay, can everyone see that?
Yeah, that's great.
All right. So Wanson and Nicole have already given awesome talks at this conference about the null ranges and Bento box packages. So in this lightning talk, what I'd like to do is use using the Bento box package, show how you can create publication quality reproducible figures with an analysis done with the match ranges package. If I happen to run out of time here, you can check out my GitHub page for the full analysis. So the purpose of null ranges is to generate a set of null hypothesis background ranges that you can use to compare your data to. So in this example, we have a granges object with 3 metadata columns, feature 1, color of each range, and the length of each range. And so what we're going to do here is we're going to define our focal set, or the set of ranges that we're interested in, as those where feature 1 is true. And the pool of background ranges to select from is going to be those that feature 1 is false. And so the goal here is that we want to use subset selection on the pool set of ranges such that they're matched distributionally to the focal set in terms of color and length. And so this is pretty easy to do with the match ranges function. We can pass our focal and pool G ranges that we've just subset into the function, along with a formula of the covariates that we're interested in matching, which in this case are color and length. And what we get out is a matched G ranges object with the 3 metadata columns that are matched to the focal group in terms of these covariates. And so what we can do now is we can use Bento box to visualize these results. With Bento box, you can define the region that you're interested in, and then use the BB page create function to create a canvas on which to display your plots. You can plot G ranges using the BB plot ranges function, where you pass your pool G ranges, for example, the region you're interested in, exactly where you'd like it placed, what size you'd like your plot, as well as the color of each G range. You can also add annotations with annotation functions like annogenome label and bbplot text. And this is the result that you get when you run this code. Since the code for plotting the focal and match sets is similar, I'll go ahead and show you what it looks like when those are plotted here. And as you can see, the ranges are in different positions, but what might be hard to see is that the color and the length of each of these are matched. So to address that, null ranges also provides a set of plotting functions that help you visualize the distributions of your match sets. So here we use the plot propensity function from the null ranges package to generate a ggplot. And then we use the Bento box bbplotgg function to place this plot exactly on the page where we want it. Again, we can annotate text labels. And since the plotting of covariates is very similar, I'll go ahead and show you what that looks like when everything's all plotted. And so you can see from this analysis that the focal and the match sets are very similar to one another in terms of both color and length. And when you're happy with your analysis and everything's done, you can use the bb page hide guide function to remove that grid and then distribute your analysis to whoever you'd like. So I'd like to thank everyone on the Null Ranges Bioconductor Slack channel who've given great input into this project. And if you have any questions, feel free to message me or Nicole or Wanson, and we'll be happy to help you get started with this package.
Thanks, Eric. That's great. Right on time. Perfect. Just remind people to pose questions in the Q&A. We'll come back at the end of the lightning talks and have a roundtable discussion, go through the questions that are there. So I'd encourage you to do that. Our next speaker is Fabricio Almeida Silva, talking about CageMiner, mining candidate genes by integrating GWAS and gene co-expression networks.
Oh, I hope everyone can see my screen. Can you see my screen?
Nope, we can see you, but not your screen. Now we can see your screen. That was perfect. There you go. That's great.
All right. So hello, everyone. My name is Fabricio Almeida Silva, and today I'm happy to present CageMiner, mining candidate genes by integrating GWAS and gene co-expression networks. So the reason why I developed this package was because GWAS can identify SNPs associated with particular traits, but they cannot identify the underlying genes controlling the phenotype. And we often have hundreds of putative candidate genes. So what should we do to address this problem? My proposed solution is CageMiner. CageMiner is a package that filters sets of candidate genes by using 3 filtering steps. In the first one, CageMiner selects all candidate genes within an interval, genomic interval, relative to each SNP. Then we select candidate genes from the output of the filtering step 1 in modules enriched in guide genes. These guide genes are genes that are known to be associated with the trait, so we kind of use them as baits. And for the step 3, we select candidates from the output of step 2 that are correlated with conditions of interest, and we calculate that with a point by serial correlation coefficient. So the input data are SNP positions as G ranges or G ranges list if you have multiple traits. Gene coordinates as G ranges also, guide genes as a character vector or a data frame, your expression data as a summarized experiment object or as a gene expression matrix, and your gene co-expression network, which you can easily import with the Bionero package, which is another package of mine, which is also in Bioconductor. Before going to the filtering steps, you can perform some exploratory analysis. For example, you can ask yourself, are SNPs evenly distributed across chromosomes? And here, by plotting this function, using this plotting function, you can see that this SNPs for this example, which is pepper resistance to Phytophthora root rot. This is a plant pathogen. We can see that SNPs are concentrated on chromosome 5. You can also do that for multiple traits, as I said, using G-ranges list objects. And you can also ask yourself, are SNPs physically close to each other? And this is something you can answer with a circus plot. And this is the plotting function. You can also plot a circus plot for multiple traits as well. And the mining steps are these functions. There are 3 functions only. Mine step 1 here. After applying step 1, we found 1,200 candidates. After applying step 2, with mine step 2, we got to 37 candidates. And after applying step 3, we got to 5 candidates only. So if you don't want to see what happens after each step, you can use the function mine candidates. This is a wrapper for the 3 previously demonstrated functions. And finally, if you want to score and rank your candidates for validation purposes, for example, you can use this formula that I developed. The score of the gene is basically the point by serial correlation coefficient times kappa, and kappa equals 2 if the gene encodes a transcription factor, equals 2 if the gene encodes is a hub in the network, and kappa equals 3 if the gene is a hub and a transcription factor. And this is what the output looks like. You have the genes and the scores. To summarize it, we have here, we have demonstrated that from 1,200 candidates, we got to 5 high confidence candidate genes. This is a reduction of more than 99%. And that's it. Here's where you can find me. And thank you very much for your attention.
Thanks, that's great. I managed to talk with my mute on. Excellent timing, way to go. The next presenter is, I should say that a couple of the presenters have posted links to their slides in the chat. I'd encourage other people to do that as well. It seems like a good way of communicating. The next presenter is Sonali Arora. Hello, Sonali, nice to see you. Variability in estimated gene expression among commonly used RNA-Seq pipelines.
Hi, everybody. I hope you guys can see my slides.
Yes.
I work as a bioinformatic analyst at Fred Hutch Cancer Research Center. And one of the things that often comes up, I work with a bunch of different collaborators, and everybody is asking me to pull up patient data from TCGA, which is a cancer repository, or from GTEx, which contains a lot of normal, healthy RNA-Seq data, and kind of just analyze it so that they can better inform the design decisions that they are going to make in the laboratory. So what I found was, when I started doing that, I found a bunch of 5 different RNA-Seq sources for TCGA and GTEx data. And obviously, I was curious about which one should I use, and what's different across each of these pipelines. I found that each one of them had reprocessed the data from scratch using FASTQ files and had applied different bioinformatic tools to get RNA-Seq abundances. They also had used different versions of the genome, different number of samples, different normalization techniques, different annotation files. And so what I did was I just downloaded the data from each of these sources. I stored them as summarized experiment objects, and then I started exploring the different data sets. So in the first figure, A and B that you see here, these are principal component analysis plots using RPKM normalized data from all the 5 different resources. And just as a reminder, the 5 different resources are GDC, which comes from the TCGA's website, Xena which comes from UCSC, VCount2, which comes from Johns Hopkins, and we have a bioconductor package for it too also. MSKCC is nothing but the CBio portal data. They have 2 versions of it. The regular normalized data as well as batch collected data, and then another one which comes from the Piccolo lab, which was probably the first TCGA resource. So coming back to the PCA plots, you can see that when I use just RPKM normalization of data along the first principal component, 2 pipelines are very different from 3 other pipelines. So VCount2 and the MSKCC pipelines are quite different. And so when I use TPM normalization of data, I see that the intervariability goes away. And if you color them in by cancer types, you can see that the variability between tissue types far exceeds the interpipeline variability for both TCGA and GTEx data. So always use TPM normalized data. The next thing that I decided to do was I decided to look at them at a per gene level. And what I wanted to do was, for the same sample, I wanted to use is check whether gene expression value was more than 32 TPM in one of the samples and less than 8 TPM. So like a 4-fold change difference in the same sample based on 2 different pipelines. And I also require that at least 1% of all samples should show that kind of difference. And I call these genes the discordant quantified genes. And these are upset plots showing the discordant genes in TCGA as well as GDEX. These are pairwise correlation plots for a couple of genes that I chose. And you can clearly see, for example, let's look at this gene called CEBPA. If you just compare recount2 and GTC, you don't, or both the pipelines give you exactly the same gene expression data for this particular gene. Whereas if you take MSKCC and GDC, there are some samples which show very different value for the same gene from 2 different pipelines. And so, the result was that we found about 12% of the protein coding genes in the genome showed different results based on which pipeline we were using. And many of these genes actually play, important roles in human tissues and various diseases in cancer and, other stuff. And the list of those genes are actually found in the supplemental table. Oops, I hear that. Okay, so all this code is actually present in the GitHub repository. And with that, I would like to thank my collaborators, Bioconductor, for making amazing packages, which makes research so easy.
Thanks, Sonali. Wonderful presentation. Our final presentation is from Jayshree Raghavan. It's a pre-recorded video, I think. It's to make the time zone a little bit more palatable. High-dimensional immune cell analysis for identification and characterization of myeloid. And then in my notes, I just have 3 more dots. So I'm not sure. Myeloid leukemia? I'm not sure. We'll find out from the slides, I guess.
I'm Jayshree Raghavan. I'm a graduate student at Indian Institute of Science. It's a great pleasure to be able to present our work on high-dimensional analysis for identification and characterization of myeloid cell subsets in diabetes. I'd like to acknowledge my advisor, Professor Siddharth Janjanwala, as well as our clinical collaborators and funding agencies for supporting us throughout this work. Various reports suggest presence of specific immune cell subtypes that have vital role in disease condition, and exploration of such remains an active area in immunology. Advent of high-dimensional analysis, coupled with their accessibility in open source platforms such as R, has made such exploration much easier to scientists. Herein in this talk, we present such exploration in chronic inflammatory status such as diabetes, where we intend to characterize different immune cell subtypes using the high-dimensional workflow established within R's bioconductor platform, which is as follows. Flow cytometry data were dimensionally reduced using algorithms such as UMAP and TSNE, and clustering was performed to visualize within this reduced dimension using algorithms such as FlowSOM and Phenograph, and these clusters were further analyzed within the established platform to look at population level differences that may exist between different disease states. Here we show that in using mouse neutrophils of both wild type and knockout animal, when dimensionality reduction such as UMAP was performed, there are no significant differences in the UMAP's islands between wild type and knockout. Further, this data was clustered using FlowSOM, and the cluster was investigated to look at which populations are different between wild type and knockout. Although there are certain trends, none of the populations were found to be significant, suggesting that there are no altered immune cell phenotypes that are unique to wild type or knockout diabetic mouse model. However, when we look at healthy human activated neutrophils and compared to diabetic as well as DFU patients, we see that the TSNE maps have distinct islands that are missing in both diabetic and DFU. Upon further investigation of flow-swarm clusters reveals that there are altered populations of neutrophils displaying altered phenotype that are of different frequencies in healthy compared to diabetic and DFU. Consistent with our previous observation, blood monocytes too upon activation showed a different phenotype in DB and DFU compared to healthy as shown in the T-SNE maps. Upon further investigation of flow-swarm clusters, it was observed that different monocyte subpopulations displaying different phenotype of various expression markers that are of lower frequency in diabetic and DFU compared to healthy individuals, suggesting heterogeneity in monocytes that are of lower frequency in diabetic population. I'd like to thus summarize that using our SpyConductor platform, we were able to show using high-dimensional analysis that no differences in blood neutrophils exist between wild-type and knockout mouse. However, when we look at healthy diabetic and DFU patients, there are differences in blood and neutrophils and monocytes, both in frequencies as well as phenotype. With that, I'd like to thank you for your attention. Look forward to your comments and suggestions. Thank you.
Thank you for the presentation, Jayashri. That's great. Those are the 6 talks, lightning talks that we had. There's a question so far in the Q&A, but I'd encourage people to post questions so we can have a lively exchange of ideas. The first question's for you, Angie. With so much data, how easy is it to identify reasonable projects?
That's a good question. I think the 5th slide in my slide deck actually explains that. I didn't have time to go into detail, but at different stage, reasonable could mean different things. For us who work in the computational biology team, reasonable projects means how do we, from GWAS signals, how do we identify those with reasonable hypothesis, with sufficient variant to gene evidence to map that GWAS signals to a actionable gene. Reasonable could mean, in this case, for example, if we have a coding signal, a coding variant that is also a GWAS signal, for example, for a disease, and that clearly links to a gene that is actionable, then that is reasonable standard in this case. Otherwise, we could use eQTL information. Oh, I didn't realize I have a
my mistake. You can keep talking. There's no
Thanks.
limit at this
Yeah,
stage.
just kidding. Yeah, but for later stage, for example, for those bench scientists, reasonable could mean different things. Like if we have a clear evidence in the mouse model, or if we have a good protein that is stable enough and it's good to develop to target that gene that we've identified. So it really means different things. But for computational biology team, there's a lot to finish there, and there's a lot of places we use R for our infrastructure.
Yeah. Can I ask, just since there's no other current Q&A, is there a tension between being a commercial company and contributing scientifically? And I mean, in part, the data there are restrictions on data use anyway, but then the derived insights, is there a pipeline for getting those exposed to the scientific community?
Not yet, but in the future, I think, yes. So in the past, we have collaborated with many academic institutions for publishing our findings with our data set, GWAS signals. In 2018, we entered this collaboration with GSK that definitely limit us a lot on what we can publish, what we cannot. But in the future, I know there are plans to publish some of the data that we are recently collecting for contributing the diversity of the genetic data to the scientific community. And also, I think, even though so far we are not publishing any of the internally developed R packages, there might be a chance in the future that we will contribute some of them to the bioconductor community as well.
Yeah, I mean, I think that's one thing that Robert championed was making the contributions that were developed at Sage and Genentech, making sure that they saw the broader community.
Yeah, definitely. Right. That's something we have kept in mind.
Yeah, yeah. Are there other questions people would like to ask in the Q&A channel? I was wondering just about RCWL, and I know that you mentioned a number of pipelines that you have. I was wondering if there's maybe a couple of pipelines that you could just highlight, that you found really useful.
Okay, sure. Yeah. Basically, like in our repertoire recipes, like for all the ICW tools, we have some basic one like SAM tools, BCF tools. It can be used for all kinds of pipelines. It's very widely used. And there are some large pipelines. For example, like for somatic mutation calling, we have integrated 7 somatic mutation caller and combined together. It's a very large project. And also some very useful pipeline like for Rna-Seq data, pre-processing to start from the fast queue to all kinds of QC and quantification and something. And we also have some very kind of latest tools like Octopus, like the latest somatic column. We follow kind of a popular tools. We try to add more, but most of the tools is kind of used in our institute, so in our center. So we focus on DNA sequencing, RNA sequencing, and single cell. But most of it are kind of a popular one. I think you can always find something useful in our repository.
Yeah. Do you use it on the local HPC or in the cloud?
In both. Like we use the ICW package to submit jobs to in our local cluster. Our local task cluster is powered by SGE, and also in the university or the UB, it's powered by Snow. Both system works very well with both actually powered by the BLC Pioneer and then the credited to Martin actually. So they works very well actually. And also we have a recently the project works on the cancer genome cloud. It's powered by SemBridge. Actually, I have to rebuild the package instead of using the SemBridge to adapt our pipelines. So basically, we can just use any tools to submit to the cloud and run it in the cloud. So basically, that's kind of like unlimited tools to run in the cloud. So,
cool. Yeah, thank you. Looks like there's some questions here. The most upvoted as directed toward Eric, and Bento box seems really cool. Have you developed a Bento box to respond to limitations in other packages like cowplot? I'm looking forward to playing with Bento box.
Yeah, that's a great question. So our philosophy behind Bento box is that we wanted the data that you plot to be in the exact dimensions that you specify. So a lot of like ggplot and other plotting devices will put your data, but also put axes and titles and annotations and legends all within that space. So this is sort of our attempt to address that, especially when you have multi-omic data like high-C plots or signal tracks that you want to line up. So the idea is to get the data to the edge of the container that you specify so that everything lines up as it should.
Cool. I think there's a question from Bianca, and she wasn't sure who she was addressing. I was wondering if the tools introduced for GWAS could be used for EWAS as well.
Yeah, I think that was for me.
Yeah.
Yeah. I haven't tried with EWAS, but I guess it should work. But I'll definitely gonna give this a try.
So unsure. And then another question from Krutik Patel for Eric. I might be wrong, but it looks like ggplot is being used for a lot of the plots shown. Can plotly or base R plots also be used in Bento Box?
Yeah, that's a great question. So we centered Bento Box to be mostly around genomic plotting, but using the bbplotgg function, any other ggplot that you use can be placed into it. Base plots, there is limited support for base plots, and some people have used it with some success, but it's not as, it doesn't usually turn out as well as using ggplots in our experience. Plotly, we have not tried, so that would be a good thing to test out. Thank you.
I guess you'd get interactivity with plotly, which could be kind of fun.
Yeah, that would be super handy, especially for genomics plots.
Yeah. I think there's a kind of related question from Aideen about Bento Box work with ggplot or a complex heat map. Complex heat map's a pretty amazing heat map plotting package.
Yeah, so any Grob-like object can be placed on a Bento Box page and relative to other plots. So as long as you can convert the complex heat map or p heat map to something that's a Grob, then it should work.
Cool. Yeah. We're almost at the end of the session. There would be time for one more question, but it looks like, so I'll take a second, give you a chance to write up that killer last question. I know that's my strategy. I don't say anything until the very end, and then I ask my killer question, which is either a total embarrassment or a good question. Usually I'm embarrassing myself. All right, looks like we're at the end of the session. It was a great, diverse set of talks. I really enjoyed them all. Thanks very much to all the speakers for the presentations, and to Erica and our AirMeet host for working behind the scenes. So a big round of applause for everyone. And I'll see you later on in the conference, including at the closing ceremonies. So see you all then. Thank you.